Greetings to everyone. Welcome back once again to my educational channel on biology. Teacher Janet here. In this video, we will study the quadrat sampling technique, which is a method to estimate the population size of plants in the school field. So, clear explanations will be given, and also we will discuss how to use the formulae to calculate population size, density, percentage coverage, and frequency. So excellent KBAT or HOTS questions will be given at the end of the video to help you practice your thinking skills and answering techniques. So this topic is quite popular for SPM, right? Especially for the objective questions and sometimes for the structured questions. The learning standards are as follows. Chapter 9, Ecosystem, 9.2, Population Ecology. After this lesson, we should be able to analyze data to estimate population size of organisms using number 1, the quadrat sampling technique, which should be discussed in this video, and number 2, the capture mark release recapture technique, CMRR technique, which will be discussed in the next video. For number 1, the quadrat sampling technique, the parameters involved in the quadrat sampling technique are A, frequency, B, density, and C, percentage coverage. So parameters are actually variables or factors that can be measured. So for each of these parameters like frequency, density, or percentage coverage, we will learn the formula on how to calculate it, and we will try out with some examples of plants and animals. So here is the concept map for this chapter. It's a very long chapter, big chapter, and we are going to discuss this, estimating the population size. All right. Now, if we enlarge this part of the concept map, the last part here, population 9.2 population ecology. So for this video, we'll be discussing how to estimate the population size by using the quadrat sampling technique, which is usually used for plants and maybe for some inactive animals, but usually for plants, all right, to determine the various factors or parameters. And then we have the capture mark release recapture technique. So this will be in the next video, so please stay tuned for this next video. And these are for animals that move. Now, what is population ecology? This is an introduction. Firstly, we'll discuss some definitions related to population ecology. What is a population? A population is a group of organisms of the same species living in the same habitat at the same time. For example, a population of grasshoppers in a field. Or as seen in this picture, a population of flamingos, which are pink birds, living near a lake or a river mouth. So population ecology is a branch of ecology which studies the interactions of a population with its surroundings. For example, the flamingos here will interact with one another and also with their environment or surroundings. They will interact with the water, the plants, and the land. Now, number 3, population size means the total number of individuals in a population. For example, 80 flamingos living in an area near the lake. Whereas population density is slightly different. Okay, population density is the average number of individuals per unit area. For example, 4 mimosa pudica plants per square meter of a field. That means for each square meter, there is an average of 4 mimosa pudica plants. All right. So to calculate population density, we'll need to use a particular formula. Estimation of population size of an organism. So two main factors measured in population ecology are A, population size, and B, population density, as we have discussed just now. So for A, population size, it is the total number of individuals present in a population. Now, the population size in an area can be estimated by a random sampling technique when the area studied is large and difficult for researchers to obtain data directly. Okay, so if the area is large, we cannot count each individual of that species one by one directly, right? So we can use a sampling technique to get samples. Okay, so let's look at this point here. A sample which represents the area is taken, for example, by using quadrats to give an estimation of the abundance and distribution pattern of this organism that inherits the area, right? Now, examples are by using the capture mark release to capture technique or by using quadrats. In both these techniques, samples are taken, right? Now, we'll discuss more of CMRR technique later, and we'll focus on quadrat sampling technique. So the quadrat is used to get a sample randomly. Now, B, population density is an average number. So it's the average number of individuals of a species per unit area. It can be per meter squared, or for smaller quadrats is per centimeter squared, as we will see with the algae called Pleurococcus species, which is a unicellular algae. Okay, but for most small plants in the field, we use a quadrat that is 1 meter by 1 meter, right? So the density will be in per meter squared. Example, 10 plants of species A per meter squared of a field. That can be the population density of a plant. Okay. Now, so first we use a number of quadrats to get the samples, and later, based on the formula, we can calculate the density, as we will discuss in the next slides. Now we'll find out more about the quadrat sampling technique. So here's the introduction. The quadrat sampling technique. What is a quadrat? A quadrat is a square frame structure made of wood, iron, or plastic, and sometimes it can be subdivided into smaller parts of the same size by using strings that are placed horizontally and vertically to divide the quadrat into smaller squares. This is used to estimate certain parameters, for example, percentage coverage of a species of plants, as we'll find out later. But sometimes, if you're just going to estimate the density, you do not need to use that. You just use a square frame to take the samples. So the quadrat sampling technique is used to estimate the population size of land plants and animals which are inactive or move slowly. Basically, it's used more for the land plants, okay, to take samples of the land plants in the habitat. Now, the number of organisms in the quadrat areas are the samples which represents the whole area of study. So an ecologist can take a number of quadrats, like 5 quadrats to 10 quadrats, and then put these quadrats randomly in the area of study. And then whatever is in the quadrat, let's say we are going to find out the density of a species of plant, which is species X. Whatever is in the quadrat, the number of the plants of species X, that will be the sample to represent the whole area of study. The data obtained from all the quadrats is used to estimate the population size and other parameters in the area of study. So the size of the quadrat used depends on the type and size of the organism, the area of study, the distribution and density of plants being studied. For example, usually in the school field, we want to estimate the population size of a plant species like Mimosa pudica, then we'll use a quadrat that's 1 meter by 1 meter, because this size is suitable for small plants like Mimosa pudica, okay, to take samples of the plants like Mimosa pudica. Okay, but if the organism is smaller, like an algae like Pleurococcus, then the size of the quadrat is even smaller. It can be like 10 cm by 10 cm in size. All right, we'll come to that later. So the size of the quadrat used depends on the plant size and so forth. Now, example, 1 meter square quadrat is suitable to estimate the population size of plants in a field. Small plants, not the very big plants, the small plants. So for a plant species being studied in the area of study, quadrat sampling technique is used to estimate 3 parameters: frequency, density, and percentage coverage of the species. So we'll be discussing all this in a short while. Now let's discuss the first parameter, which is frequency. The first parameter or factor is frequency. Frequency is the probability of getting an individual of a particular plant species in each quadrat. For example, out of a total number of quadrats, like 5 quadrats, how many contain the plant species? Whether it's one individual or more, doesn't matter, as long as there's at least one. Okay? So the frequency equals to the number of quadrats containing the plant species divided by the total number of quadrats used times 100% to convert it into a percentage. So the frequency is a percentage, right? Now, so let's look at this example here. Here there are 4 quadrats used, and quadrat 1 contained 2 of the plants of a particular species. Quadrat 2, none. Quadrat 3, there are 2 plants, and quadrat 4, there are 3 plants. So in the exam, you can be given a picture like this, okay, with the quadrat already placed in the school field, and then you can be asked to jot down, determine how many plants there are in each quadrat. Okay, quadrat 1, 2 plants, quadrat 2, 0, quadrat 3, 2, and quadrat 4, 3 plants, right? Now after that, you can be asked to calculate the frequency and density, okay, for structured question. So try and calculate the frequency. Take out a pen and paper and calculate the frequency based on the formula given here. So what is your answer? Did you get 75%? So the percentage frequency of frequency equals to 3 over 4 times 100%. That means out of these 4 quadrats, 3 contain the plants, right? Quadrat 1, 3, and 4 have at least 1 plant, right? And only quadrat 2 has 0 plants, right? So 3 out of 4 have the plants, so you multiply by 100% to get a percentage. That is 75%. The frequency is 75%. Next, let's discuss density. So density is the average number of individuals of a particular species in per unit area of study. For example, 7 individuals or 7 Mimosa pudica plants per meter squared on the average. Okay, so density equals to, now this is the formula, total number of individuals. So it's now a number of individuals of the species in all quadrats. Okay, number again is important, over the total number of quadrats used times the area of 1 quadrat. So assuming that the area of 1 quadrat is 1 meter squared, all right, can you calculate the density based on the results here? So try it yourself first, and then we'll discuss in a short while. Right, so what's your answer? Density equals to 9 individuals per meter squared. Let's see how to calculate that. Now, density equals to total number of individuals of a species in all quadrats. So usually you see all these numbers, you have to add it up together, all right? And then this is the total number of individuals of the species, like the Mimosa pudica plant, in all the quadrats. You have totaled it up, divide by the total number of quadrats. So there are 5 quadrats here, all right? 5. Even if one quadrat has 0, the reading is 0 number of the plant, you still have to count it in, okay, even if it's 0. But in this case, all of them have the plant, okay? But anyway, you use 5 quadrats, you have to write down. Divide by 5 times, in this case, times 1 meter squared, because that's the area of 1 quadrant, okay? So your answer will be 9. 9 what? 9 individuals, or 9 of these Mimosa pudica plants. 9 plants per meter squared, okay? So take note of the unit, individuals per meter squared. Now, next is the percentage coverage, which is slightly different. Coverage, the word coverage means the surface area of the soil which is covered by the shoots or leaves of the plant species, okay, by the leaves especially. So here you can see let's say for this grass, what is meant by coverage? It is the area below the grass that is covered by it, the soil. The area of the soil is covered by this plant when you look from the top, right? So that's the coverage, and then from there you calculate the percentage coverage, which is the percentage of soil surface covered by the plants, the percentage, okay? So, for example, for a species of grass, now percentage coverage equals to total area. Now it's not a number, it's the area covered. It's not the number of the individuals, okay? It's the total area covered by a species in all quadrats per the total number of quadrats used times the area of one quadrat times 100%, okay? So, for example, okay, let's look at our results here in this picture. Okay, here you can note the number of quadrats used and assume that the area of one quadrat is 1 meter squared. So now you're going to find or calculate the percentage coverage based on the results here. All right. So can you try to calculate the percentage coverage based on the results here? Right, let's look at the answer. The percentage coverage is 50%. Did you get this answer? Okay, let's see how to calculate percentage coverage. Add up all the coverage or area that's covered by each quadrant. Okay, area that's covered in each quadrant, area that's covered by the plant species in each quadrant. Okay, so 0.8 plus 0.7 plus 0.3 plus 0.2 meters squared. Then divide by the total number of quadrats. Now take note there are only 4 quadrats used here, as you can count 1, 2, 3, 4. Okay, times the area of one quadrat. Okay, so times 1 below here, and then times 100%. Right? Answer is 50%. That means here you can say that overall 50% of that field is covered by the grass species. Okay. So in this section, we are going to discuss how to carry out field studies using the quadrat sampling technique to determine the density and the percentage coverage of a species of plant, as shown in this diagrams here. So this is related to activity 9.3 and activity 9.5 in your KSSM textbook. Now let's discuss how to carry out a field study using the quadrats. This is activity 9.3 in your textbook. The aim is to carry out a field study to estimate the population size of a plant species in the school field. For example, the plant species can be Mimosa pudica plant, okay, that closes its leaves very quickly when we touch it, right? So apparatus used is a quadrat or more quadrats, maybe 10 quadrats measuring 1 meter by 1 meter, a pen and a notebook. Okay, if you use one quadrat, that means you have to do the experiment, repeat the experiment with the same quadrat, right? Now the procedure. Identify a plant species X present in the school field, as we said, for example, Mimosa pudica plant, and we have to know how to identify it well. Then a quadrat measuring 1 meter by 1 meter is randomly placed in part of the school field. So randomly placed, that means you don't plan that to place it at a specific spot. You just put it there, okay, quickly and without preparation. And in Malay we say secara rawak. All right, this is very important for the field study to be accurate and successful. Right, after you place the quadrat in the school field, you count the number of individuals of that species X in the quadrat. Now repeat steps 2 and 3 at 9 other sites. Maybe you can still use the same quadrat if you want, okay? And at 9 other sites of the field randomly selected also for the same plant species. Okay, so this diagram here shows you the results, okay, of the 10 quadrats that were used and the number of plants in each quadrat. Like quadrat 1 has 4 of the plants of that same species, species X, right? Quadrat 2 does not have any of this of the individuals of this plant species, and so forth. Record the results in the table, so tabulate your results. Calculate the frequency, density, and percentage coverage of species X. Now, if we are calculating percentage coverage, we have to find the area covered by the plant in each quadrant first. That means you have to divide the quadrant up using strings. Okay, divide it into smaller squares. All right. And then for density and percentage frequency, we don't find the area, we count the number of plants in each quadrant. So here's the table with the tabulation of results. Plant species X, number of individuals in each quadrant. For example, quadrant 1, there are 4 individuals. Quadrant 2, there are 7 individuals of species X, and so forth. So normally, we will have to total up the number of individuals, right? So 4 plus 7 plus 2 plus 3, and so forth, you get 40 individuals. Now, you're asked to find the density of species X in per meter squared. So try and answer this question first. Write down your working, show your working, and then give the answer in this column here. Right, so did you get this answer? 4 individuals per meter squared. So this is not a percentage, it is individuals per meter squared. Now, based on the formula given here, density equals the total number of individuals of a species in all quadrats, divided by total number of quadrats used, times the area of one quadrant. Remember, you don't have to times by 100%, okay? Then you will get this working. 40 is the total number of individuals of the species X in all the quadrats. Divided by total number of quadrats, we use 10 quadrats, so 10 times the area of one quadrant is 1 meter squared, because the quadrant has the size 1 meter by 1 meter. So answer is 4 individuals per meter squared. All right. Here I write down the units again, but it's already in here, okay, in the table, in the title. How about frequency? So frequency, it's very easy to calculate frequency. Just find out which quadrant has 0 number of the plant species. All right. So there's one quadrant, right? Quadrant 9. That means out of 10 quadrats, 9 quadrats have the species. So 9 over 10 times 100, you get 90%. Next, we want to calculate the coverage and then the percentage coverage of a plant in the field. All right. So how to determine and calculate coverage of a plant X in the quadrant? So this is something optional, and it's going a bit more into detail, okay, but you'll need to know this if you are really doing the field study, right? So let's say the quadrant is 1 meter squared, 1 meter by 1 meter here, or 1 meter equals to 100 centimeters. Now, if you want to calculate the coverage, that means the area covered by the plant, for example, this is the plant, it covers the area of the school field. Usually this type of plants that where you can calculate coverage, they must be plants that grow very close to the soil, to the ground, okay, like a grass, a type of grass that creeps on the ground, right? So to calculate the coverage, you first have to divide the quadrant into smaller squares so that we can count the number of tiny squares that have the plant, okay, when you put the quadrant on top. So we can divide it into 100 small squares here by using strings to divide it, all right? So every 10 cm, you tie a string from one end to the other using nails and the string to tie. Now every 10 cm, you tie another string, vertically and also horizontally, okay? So in that way, you divide up the quadrant into 100 small squares. Each small square has the size 10 cm by 10 cm, or 0.1 meter by 0.1 meter. So the area of one tiny square equals to 0.1 meter times 0.1 meter is 0.01 meter squared. Okay, so this is the size of one small square here. Now, next, after you have placed the quadrant on top of the plant, you will see that certain parts of the quadrant is covered by this plant, right? So you need to count the number of squares that contain the plant. All right. Now to do that, you only count squares which are half covered or more than half covered. If the square contains a bit of the plant, like at this point here only, not even half covered, you just ignore it and don't count it as one square. Okay, so if I'm going to count the number of squares for this quadrat that is occupied by the plant, covered by the plant, okay, the area is covered by the plant. So let's have a look. How many squares are there here that you can count? So there's 1, 2, 3, 4, 5, 6, 7, 8, 9. This one can be counted in, so 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Okay, the rest here, they don't, the plant doesn't even cover half the square, so they are not counted in. Okay, so there are 20 squares with the plant. Now each square is of the area 0.01 meter squared, right? So 20 squares times 0.01 meter squared. This is the area covered by the plant X, or the coverage. 20 squares times the area of 1 square, which is 0.01 meter squared. Answer is 0.2 meter squared. Okay, just use your calculator and calculate. All right, so this is how we calculate the coverage of the plant. And from there, we will calculate the percentage coverage. Right, so now let's look at the tabulation of the data and then also how to calculate the percentage coverage. So plant species Y, the quadrat, okay, is shown here, and then the coverage is below here, okay, all the values here. For example, quadrat 1, there's an area of 0.5 meters squared that's covered by the plant, right? So after you have tabulated the data, then you have to calculate the total area covered by species Y in meters squared. So normally you will have to add up all the values here. Okay, so 0.5 plus 0.4 and so forth, you get 3.7 meters squared. That's the total area covered by species Y in all the quadrats. So what is the percentage coverage in percent? Try and calculate this yourself first, and we'll look at the answer in a short while. Right, so did you get this answer? 37%, right? So as usual, if we look at the formula, we follow the formula here. Percentage coverage equals to the total area covered by the species in all the quadrats, which is 3.7, divided by the total number of quadrats, which is 10, and then area of one quadrat is 1 meter squared. Okay, so 3.7 divided by 10 times 1. Then you have to multiply by 100 to change it into a percentage. So the answer is 37%. Okay, so this picture here shows you a example of a quadrat with the area covered by the plant species. 0.3 meters squared of this area of the quadrat area is covered by the plant species. Let's go on to activity 9.5 in the textbook. So we are going to estimate the population size of Pleurococcus species. Okay, in this activity, introduction on Pleurococcus species. It is not a plant. It is a tiny unicellular organism and is classified as a type of green algae. The structure. The cell is spherical or rounded in shape, as you can see in this picture here, and also in this photo here. And it has a thick cell wall, okay, as shown in this diagram. And you can see here the thick cell wall, which helps to prevent excessive water loss. Okay, but it's still sensitive to heat and bright light. With a nucleus and green chloroplast to carry out photosynthesis. So the cell is green in color because it has the green chloroplast. So it's an autotrophic organism that can carry out photosynthesis and produce its own food. But it grows on the tree trunks to have a surface that is high enough to obtain sunlight for photosynthesis. They exist as single cells or live in colonies as green patches. Okay, like colonies are where a lot of the cells are attached to each other. Okay, and then they form green patches on the tree trunks. Reproduction is vegetative, that means it's asexual reproduction, through cell division by binary fission, where one cell divides into two, and two cells into four, and so forth. Something like bacteria, okay, that carries out binary fission. Now, habitat. Habitat means the place where it lives. It's the moist bark of trees, rocks, and moist soil. There must be some moisture for it to survive. Favorable conditions, they will grow in areas with some sunlight, but they must be protected from the hot, scorching sun, right? So they try to avoid dehydration. They can only grow in areas that have some sunlight, moderate sunlight also. So here's the procedure to determine the percentage coverage of Pleurococcus species, or the coverage of Pleurococcus species. Number one, tie a string round the tree trunk at a distance of 1 meter from the ground. Okay, the string is tied at a distance of 1 meter, the blue string here, from the ground. So to fix the height at which we are going to do our field studies. So all the transparent quadrats will be placed at 1 meter from the ground on the tree. So number two, draw grids measuring 1 cm by 1 cm on each of the transparent quadrats of size 10 cm by 10 cm. So the quadrat used here is very small. It's only 10 cm by 10 cm. And you have to divide it further into smaller squares by drawing lines that are 1 cm apart from each other. Okay, so you can draw lines vertically 1 cm apart from each other and also horizontally. So in that way, you divide the quadrat up into 100 small squares in order to find the coverage and percentage coverage. Next, put the quadrats at the string on the tree at different aspects or directions, P, Q, R, S, T, as shown here, P, Q, R, S, T, and this is to find the coverage at different aspects and relate them to the abiotic factors at different aspects. The light intensity and temperature will be different for different aspects. So by using a marker pen, shade all the parts where the growth of the green Pleurococcus species can be seen in the transparent quadrats. For example, let's look at this picture here. So we can see that the green Pleurococcus is growing at this part here, and the transparent quadrat is placed on top of it, right? So you can either draw the outline of the green patches here, which is the Pleurococcus, or you can shade the parts of the Pleurococcus by using another marker pen, maybe a green one, green ink, right? So after that, you have to determine the coverage. To determine the coverage in cm squared, count the number of squares which contain Pleurococcus species in each quadrat. Take note that we only count squares which are half covered or more than half covered. Okay, as like before. For example, this square has just a little bit of Pleurococcus, about a quarter part of the square, so it's not counted in the field study. Okay, so if you count the number of squares here, you should get 49 squares, which is equivalent to 49 times 1 cm squared. That means 49 cm squared. Each small square is 1 cm squared in terms of its area. Now, lastly, calculate percentage coverage by using this formula. So we have to know the individual coverage for each quadrat first. Okay, then if we want to generally find out what's the percentage coverage of the Pleurococcus on the tree, on this tree, then we can use this formula. Total area covered by Pleurococcus species in the 5 quadrats, generally in the 5 quadrats. So this is for one quadrat near 49 cm squared, but there are 4 more quadrats. You add up all the areas covered by the Pleurococcus and then divide by total number of quadrats used. In this case, it's 5 times area of 1 quadrat is 100 cm because it's 10 times 10 cm squared. Okay, and then times 100%. That is your percentage coverage. Okay, and then one more thing is in certain SPM questions, they ask for percentage coverage of bread mold on bread. So the procedure is similar. We place a quadrat on top of the bread mold, which may be the same size like the quadrat, okay? And then we just count the number of squares, like in this case, the number of squares that contain the bread mold, okay? And then to find the percentage coverage for just one piece, okay, you have to take the area of coverage for the bread mold, like for example this green bread mold, and then you have to divide by the total area of the bread, and then times 100%. You'll find out more about this when we look at the last question that is in this video. So in the textbook, activity 9.5, the aim is to carry out a field study to observe the effects of abiotic factors on the population of Pleurococcus species. Abiotic factors here are the physical factors like light intensity, temperature, and moisture, which may vary for the different aspects or directions. Number 1, place the quadrats on different sides of the tree trunk, north, south, east, and west. All right, that means facing for different aspects or different directions. So from P to Q to R to S, T, and T. Now, for each quadrat location, record the abiotic factors. For example, light intensity is measured with a light meter, temperature is measured with a thermometer, and moisture levels are observed. So number 3, after that, you calculate the percentage coverage or the coverage of Pleurococcus species for each quadrat. Use a marker pen to shade all the squares within the quadrat that show the growth of Pleurococcus species, and then you count the number of shaded squares. In this case, you don't even have to know how many cm squared it is. All right, just count the number of squares and then calculate the percentage coverage for each quadrat. That means in this case it's for one quadrat only, right? Not for all the quadrats. For one quadrat, the percentage, sometimes it's called percentage coverage or just coverage, is the number of shaded squares per the total number of squares in the quadrat. So maybe your quadrat has 100 squares, right? So you count the number of shaded squares, it may be 45 or 50. So 50 divided by 100 times 100%. That means the percentage coverage or just coverage is 50%, okay, for that quadrant. Then for each quadrat, you correlate the growth of the Pleurococcus and the percentage coverage or coverage with the abiotic factors to find out what are the suitable, favorable abiotic factors, the levels of the abiotic factors that are suitable or favorable for the growth of Pleurococcus. So conclusion is that favorable abiotic factors or conditions for Pleurococcus growth are the following. Number 1, moderate light intensity for photosynthesis. Number 2, sufficient moisture to prevent drying. Number 3, moderate temperatures for optimal photosynthesis and hydration so that the Pleurococcus doesn't dry out. And it needs a moist environment like shady, damp areas with some amount of sunlight for photosynthesis. Okay. But high light intensity and temperatures can cause Pleurococcus species cells to lose water and become dehydrated. So in the aspects that have high light intensity and temperatures, the growth of Pleurococcus will be low. Okay, the growth rate will be low, and there'll be less coverage. Now let's test ourselves by trying some questions that are SPM standard, and also there are hot questions for you to exercise your thinking skills. Question 1. A student carried out a field study to determine the density of a small herbal plant, Z, growing in the school field. Okay, for example, like something like this plant. He used quadrats of size 1 meter by 1 meter. So the quadrats are shown here with the number of the plant Z found in them. Right? Now, this is a structured question. Based on the quadrat sampling results as shown above, what is the estimated density of plant Z? Show your working. So density, highlight this word. You want the density. So can you remember the formula and then try this question yourself first, the density, and then what is the percentage frequency of plant Z? Okay. So 1 mark for working and 1 mark for the answer for both questions. Sometimes it's only 1 mark. If it's only 1 mark, you still have to show your working. And also remember to state the units for your results. Okay, let's look at the answers. Now, density actually equals to the total number of the individuals, right, in all the quadrats. So you have to add up the number of plant Z in all the quadrats over the area of one quadrat, which is 1 meter by 1 meter squared. So it's 1 meter squared. and the total number of quadrats, which is 5, correct? So 14 over 5. And then the answer is 2.8. Now 2.8 is not enough, you must have the units. Individuals of plant Z, okay? Individuals of plant Z per meter squared. The unit is plants per meter squared. Alright? So without the unit is also wrong. Now, what is the percentage frequency of plant Z? Percentage frequency. So try and recall the formula for percentage frequency. Alright, let's have a look. Now, percentage frequency equals to the number of quadrats that contain the plant, which is 4, right? Out of 5, 4 have the plant. Quadrat number 2 doesn't have the plant. So over the total number of the quadrats, which is 5, times 100%. So the answer is 80%. Take note here that percentage frequency of frequency, the unit is percent. All right. Question 2. An ecologist wanted to estimate the population of water lettuce or Pistia species in Lake R. The total surface area of Lake R is 400 meters squared. He used quadrats of size 1 meter by 1 meter to carry out the random sampling in the lake. The results are shown below. Okay. So what is the estimated population size of Pistia species in Lake R? Try this question first and we'll discuss it in a short while. Right, what is your answer? The answer is B. Okay, so there are two methods of doing this calculating and getting the answer for this question. The first one is just by using common sense, calculate using proportion. So method A is like that. Total number of plants in the 5 quadrats with area 5 meters squared. Now each quadrat is 1 meter by 1 meter, so the area is 1 meter squared. But you have 5 quadrats, so your total area for all the quadrats is 5 meters squared. Okay, but first you have to add up all the numbers here. Usually you always have to add up the numbers. So 9 plus 10 plus 18 plus 24 gives you 61 plants. So in 5 meters squared area, that means in the area for 5 quadrats, you have 61 plants in total. So use proportion. That means 5 meters squared area gives you 61 plants. So how about an area of 400 meters squared, which is the area, total surface area of the lake? So by proportion, it should be 400 divided by 5 times 61, and that will give you 4880 plants. Okay, because you know the area of the lake, so you can find the population of the Pistia species in the lake. Now, another method is to calculate the density first. So we know the formula for density, right? So density is actually the average number of plants per meter squared. So density of Pistia species in the lake equals to total number of individuals in all the quadrats divided by the total number of quadrats divided by the area of one quadrat also. Okay, so total number of individuals in all the quadrats. So again, you add up. This one you add up all the numbers, you get 61 plants, right? Then the total number of quadrats is 5, and the area of one quadrat is 1. So it's actually 61 divided by 5, and you get 12.2 plants per meter squared. That means in every meter squared, you have 12.2 plants. Okay, now you multiply by the total lake area, which is 400 meters squared. So if you have 12.2 plants for every meter, 1 meter you have 12.2 plants, 1 meter squared. For 1 meter squared, you have 12.2 plants. So in an area of 400 meters squared, how many plants will that be by proportion? So 400 times the density. Okay, the area, total area times density will give you the number of plants, total number of plants, or the population of Pistia species, which is 4880 plants also. Question 3. The diagram shows a field study carried out to investigate the distribution of a green algae, Pleurococcus species, on a tree trunk. The size of each quadrat was 10 cm by 10 cm, and the following results were obtained. So what is the percentage coverage of Pleurococcus species on the tree trunk? Try this question first by yourself. Right, what's your answer? B again, 36.7%. So let's see how we obtain this answer. percentage coverage, right? So we have to use the formula percentage coverage equals to total area covered by a species in all quadrats divided by total number of quadrats used, and also the whole thing divided by area of one quadrat, and then you have to multiply by 100%. Okay, so in this case, what is the total area covered by the Pleurococcus? So again, we have to add up all the values from the quadrats. So the numbers from the quadrats. Now this is the coverage. So add up all the coverage, or the area of coverage. 20 plus 25 plus 80 plus 33 plus 62. Even the zero, I mean, we have to include this quadrat in as one quadrat, even though there's zero Pleurococcus there, no Pleurococcus there. So anyway, divided by, so the number of quadrats is not 5, it's 6. 1, 2, 3, 4, 5, 6, including the one with no Pleurococcus. Then this one is multiplied by area of one quadrat, as we know from here, is 10 by 10, so it's 100 cm squared. Okay, so then multiplied by 100%. Okay, so coverage is in cm squared also. Now the answer is 36.7%. Okay, calculate, use your calculator. Question 4, the last question. A piece of moist bread was kept in a cool, dark room. The diagram shows the distribution of green mold and black mold on the bread after 10 days. What is the percentage coverage of green mold on the bread? So try this question first, and then we'll discuss the answer. Right, what is your answer? 42%. Okay, so let's see how to get this answer. First, there's only one quadrat here, so the percentage coverage is only for one quadrat here. So first find out the total area of the bread. That means it's the same size as the quadrat, in this case, almost the same size. Now here is written 5 cm, this quadrat is 5 cm in width. So what about the length? So you count the number of squares here, because one side of the square here is 1 cm in length. So 1, 2, 3, and so forth. You count, there will be 10 squares here. Okay, so that's 10 cm. So the total area of bread is 5 times 10 equals to 50 cm squared. Next, find the coverage or area of coverage of the green mold. Okay, so count the number of squares here, but only count squares which are half covered or more than half covered. So if the square is only like a quarter covered, then you have to ignore it. Don't count it as one square. Okay, so if you count the number of squares here, it should be 21 squares. All right, or 21 cm squared, because one square has an area of 1 cm times 1 cm. Okay, as shown here. So 21 cm squared. So percentage coverage is very easy. It's just the total area of the bread mold divided, the area of the bread mold divided by the total area of the bread. Okay, area of bread mold divided by total area of bread. Okay, so area of the bread mold here divided by the total area of the bread, which is this size, same size as the quadrat. So times 100%, and you get 42%. Okay, so quite simple, but only one quadrat is involved, not many quadrats like what we have discussed earlier. Right, that's all for this lesson. I hope that you have benefited a lot from this, and that you have tried all the questions to gain more confidence in doing the calculations. So please do share, like, and subscribe to my channel, and comment below the video. Till we meet again, goodbye.